record it. All right, so let's look at McGraw-Hill again just to be clear about the anatomy resources. So we go down here to our cadaver thing, and it's way more than just a cadaver, the virtual dissection. So, for example, if we go back into skeletal, and obviously you can look at individual bones. So I do like this. I know this is not what we were talking about, but I just want to point out that the bones that are hard to visualize in real lab, like that is a beautiful ethmoid bone right there. And most of the things that we were trying to find are on there, right? So like middle nasal conky. I don't remember which ones I put on your list, I'll be honest with you. Perpendicular plate, right? And then like the crystal galley up there. So I do love that. But if you go to the microscope one, now instead of selecting dissection stuff, you're in the histology section. So there's going to be a histology section for every system. And for the skeletal system one, of course, the histology options are the different types of cartilage and bone. So we get 2 different magnifications of elastic cartilage. That is a beautiful slide right there. This low one is a really horrible picture. I don't know why they used it when beautiful things like that, like there is no magnification difference there. I don't know what's going on, but anyway, you don't need to know the individual things, but it can be helpful to, you know, select your different parts and say, oh, look, those are the chondrocytes and the lacunae, and here's the elastic fibers. So pointing this out in part because as we go forward, this might be useful for different slides. And then like here's our beautiful bone slide that we've seen before that we got to look at in real life. Come on, there it is, right? Beautiful. And then if we go to muscular, and you'll notice it resets to dissection, but we go back to the microscope, now we're in histology, and we can look at smooth muscle, cardiac muscle, skeletal muscle. They've even got the sarcomere. So this is very similar to the picture that I have in your guys' lecture. You don't need to look at it for lab. We're not doing this in lab, but I'm just pointing out that it's there. And that you can, you know, look at this stuff. So A-band, I-band, C-disc, like that's pretty cool. But anyway, so we've got cardiac muscle, for example, and highlighting the intercalated discs and kind of showing you the branching nature. And it's good to know the nuclei, I guess. And then there's the blue one, just like we had in lab as well. So the point of this one is, of course, to highlight the intercalated discs. And then we can look at things like the smooth muscle. And this is low magnification, showing you the smooth muscle, mostly in longitudinal section. And then this one is nice because it highlights that fusiform shape of the smooth muscle. But again, for our purposes, we just need to be able to identify the type of tissue, right? It's just tissue type. That's all we're focusing on for what we looked at. So you're like, cool, that's smooth muscle. And then skeletal muscle, right? Really long and striated. So we do have that in this exact same resource as we have in the dissection. And this still isn't going to work. This is the 3D modeling one, but my internet is not the best quality, so this is not working. Okay, that's fine. Don't do it. Don't do it. Don't worry about that. Anyway, if it works for you, cool. But you want to focus on the dissection one for the most part. Other questions? So like I said, there's no new lab stuff for this week. It's just continuing to do what we already did. And we can tackle the details of the exam formatting and stuff on Wednesday. But my plan is to try to make it as close to what it would have been live as possible. And I will say, I think there is one advantage to this because I can screenshot the different, ooh, cool. Sorry, the 3D thing just worked. Here, look, if you haven't tried it, it's interesting. Ooh. Probably not quite as good as the other thing we had, but you know. And it looks like all we have is head and neck, so that's cool. But anyway, what did I say? Oh yeah, so the nice thing about this is that because I can potentially screenshot this stuff with the highlights, you'll always know exactly what I'm asking you about. And I know sometimes that gets confusing when I have the models and the stickers or the pins or whatever it is, you know. But with this, it should be very unambiguous because you're going to be able to see exactly what is being asked about, you know, like no question, like which one is highlighted. So hopefully that will actually help with the muscle and bone stuff. Because we can do that for the bone too. So for example, right, so here's a nice hand. And like I said when we were live, I'm not going to give you individual bones, right? It's always going to be all connected so you can figure it out. But you'll know that that's the bone I'm asking about, right? Because now it's a different color. Or it can be this one. And so this is a nice way to study without getting confused about it. And it's also going to be a nice way to tell exactly which one you're being asked about. Because it, you know, like there's the radius. I'm not going to ask you the radius on this picture. That's stupid. But yeah, so I think I think it'll be relatively straightforward as long as you're comfortable with what's on the list. So again, if you have any other lab questions, we can tackle them on Wednesday. If you've got one right now, I can answer it, but if we can definitely refocus on that on Wednesday, because you know right now we've got the lecture test to get through first. How do you get to the lever system? What do you mean? Like, where is it in the lectures?
I thought you mentioned to, where you said review the lever system, but I don't know if you meant to do it on this, the thing you were going over, the online with the pictures. Is there anything like that? Or is it just in the PowerPoint?
No, it's just in the PowerPoint.
Okay.
Yeah. I think so. Okay, any other questions right now? Okay, you can email me, of course, anytime. I will try really hard to get back as soon as possible right now, since we've got the test. So I'm assuming if you're going to email me in the next 24 hours or so, it's going to be about the test. So I will do my best to keep a close eye on that. And then, yeah, on Wednesday, we'll be back starting new material and lecture and available to keep talking about the lab stuff as needed. Okay.
Okay. Thank you.